In this video, we will discuss about the modifications of plasma membrane. The first type of modification of plasma membrane is microvilli. Microvilli are finger-like projections which emerge out of the cytoplasm in the form of microvilli. So this is the cell of renal tubule which bears microvilli, and microvilli are also found in our gut mucosa, in the mucosa of intestine or intestine. These increase the surface area for reabsorption or absorption. Microvilli are supported by microfilament form of actin, and this actin is attached anteriorly to the microvilli with the help of alpha actinin. This actin is also attached to the plasma membrane with the help of calmodulin, and at the base this is forming a network of actin and myosin which collectively forms the terminal web, and there is cytoplasm in the core of microvilli, and there is plasma membrane which has a fuzzy coat of glycocalyx on the surface. This is microvilli. This increases the surface area for absorption or reabsorption. Absorption in case of intestinal cells and reabsorption in case of renal tubular cells. Renal tubular cell is found in the renal tubule. For example, if this is a nephron, then it has two parts. One is renal corpuscle, another is renal tubule. This is renal tubule, and the cells have brushed borders formed by microvilli. This brush border increases the surface area for reabsorption of fluids, and here in the glomerulus filtration takes place, and here reabsorption takes place. So this cell comes from here, from renal tubule. Next to microvilli is junctional complexes. There are two types of junctional complexes. One is interdigitation and another one is tight junctions. So next type of modification of plasma membrane is junctional complexes. First is interdigitation. Interdigitation is simply a junction where two cells make folds in such a manner that they look like digits which are interdigitating each other. This is interdigitation. This is formed in order to increase the surface area for exchange of molecules. This increases the surface area for exchange between two adjacent cells. Another type of junctional complexes is tight junctions. Tight junctions are formed by close association of two cells between their plasma membrane, and they are found in the same tubular cells, renal tubule cells. This is one tubular cell, renal tubular cell, and this one is another. So this is the apical membrane and this is basolateral membrane. Here at the start of basolateral membrane, tight junctions are formed. In case of tight junctions, there is no gap between the two plasma membranes of the cells, and these junctions are stabilized by sealing strands. In between the plasma membranes, sealing strands are there, and there are membrane proteins which make a tight coordination with each other, and these also form contact with the cytoskeletal components. This is the plasma membrane, these are membrane proteins, and these are filaments of cytoskeleton, and in between the two membranes, sealing strand is present. These are tight junctions. This does not allow the movement of anything across them. Next type of junctional complex is desmosomes. So there are several types of junctional complexes. The third one is desmosome, A, B, and then C. Desmosomes are of two types. One is belt desmosome, and another one is spot desmosome. Suppose this is one cell and this is another cell, then desmosomes are formed in such a manner that the protein from one cell, membrane protein of one cell, makes contact with another protein from another cell, and interiorly they are attached to a belt of strands in such a manner that this belt makes contact with other nearby or neighboring cells, and this is belt desmosome. And in case of this belt desmosome, the intercellular space is sealed with cementing material which is formed of alpha actin, and this belt is also formed of actin. So this is belt desmosome. Second is spot desmosome. In case of spot desmosome, there are two cells. Their membrane proteins are making connections. They are holding each other, and their cytosolic end is associated with disc-like fibrils, which are tonofibrils formed of keratin, and also cilium material is there in between the spotodesmosomes. So there is disc-shaped material, disc-shaped cytoskeletal component which supports the cell membrane proteins which are forming junctions. So next is D. D is hemidesmosomes. Hemidesmosomes, these do not have tonofibrils, but instead of tonofibrils or keratin, the hemidesmosomes, this is cell 1, cell 2, and this is membrane protein which is forming association with the surface protein or membrane protein of another cell. These make association with the collagenous strands which are present inside the cells. This strand is formed of collagen. Hemidesmosomes are found in the intercalated discs between the cardiac muscle cells. Next is gap junctions. Gap junctions are formed between two cells which allow free passage of molecules between the cytoplasm of the two cells. Gap junctions are formed like tunnels. For example, this is cell 1, and from this cell arise a tunnel which is formed of 6 subunits, 1, 2, 3, 4, 5 and 6 subunits, and another cell also has same tunnel which is formed of 6 subunits, and these have a diameter of 20 angstrom. This tunnel has diameter of 20 angstrom, so this allows free passage of small molecules. This is gap junction. Gap junctions are found in the intercalated discs of cardiac muscles. This region is intercalated discs, and the cardiac muscle cells are associated with each other through intercalated discs where desmosomes and gap junctions are present. Gap junctions allow free entry of sodium ions. When one cardiac muscle fiber gets the action potential, the signal of action potential, then that wave is spread in the entire atrium or entire ventricle. So the atria or the ventricles, at one time all the atrial muscle fibers contract at once, and at another time all the ventricle or ventricular muscle fibers contract at once, because they are connected with gap junctions. So these are the modifications of plasma membrane.